Hi folks, and welcome to this Python for Biologists video. Today we're going to be taking a fairly detailed look at a question that often arises in courses and emails and general correspondence with bioinformatics folks, which is to do with making Manhattan plots, and specifically making them in Python, and even more specifically making them with the set of tools that I normally like to talk about, that is to say pandas, seaborn, matplotlib, numpy and so on, the usual scientific Python stack. I'm hoping with these videos to do something a little bit different from my normal articles. I've been wrapping up work on doing a bunch of stuff for a biological data visualization book, and I've ended up with a bunch of examples that, although really nice, didn't really fit anywhere in the book. So I'm going to try and turn these into videos, and we'll be able to take a more in-depth look at a few topics that then could fit in the book, like I say. So the question of how to make a Manhattan plot in one sense is kind of boring, right? It's just a scatter plot, but it actually, when we dig into it, turns out to have some interesting aspects to it. It touches on a bunch of topics to do with the interaction between the different scientific libraries that we use in Python, so things like pandas and matplotlib. It touches on topics of why we might use pandas to do this rather than other tools, because there are plenty of tools that exist that will draw Manhattan plots. And there are even a few hints about data visualizations in there, and I'll be able to sprinkle in a few useful tips and techniques. So before I get rid of the video of my face and we switch to screencasting, a couple of housekeeping notes. As you can probably see in the background here of this window, I'm going to be working a Jupyter notebook, and I'll make sure to put a link to the final notebook in the video description just underneath. So don't try to pause the video and copy and paste and so on. All of the code that I type in is going to be in a Jupyter notebook on GitHub, and I'll put a link there so you can just click through and copy and use any of that code if you like. I'm going to be using quite a lot of different seaborn and pandas and matplotlib techniques, so we're not going to try and explain everything in a super lot of detail. That would make the video many hours long, but all the methods and functions and so on are all described in the biological data exploration book. So if you're interested, I'll put a link in that in the exercise description as well. And one final note is that I've recorded this in high definition, so if you want to be able to see the code and the visualizations comfortably, you probably want to watch this full screen unless you're on an enormous monitor anyway. Hopefully I'll be able to explain a few things in this video, but if anything isn't clear, then do go ahead and ask questions and so on in the comments. Okay, let's jump in. I'm going to just put the Jupyter notebook here full screen, and you'll see I've opened a brand new empty notebook. So what actually is a Manhattan plot? Let's start off with that. And I'm actually going to go immediately to Wikipedia here, because Wikipedia has this nice picture, which hopefully you recognize if you're watching this video. I'm guessing you have at least some interest in Manhattan plots, and so you'll recognize this is the kind of thing that we're trying to make. And so this is a type of plot that we've come up with in bioinformatics to display the association between SNPs at various positions in a chromosome. Given that there are 22 here, we might guess that this is probably a human genome we're looking at. So the SNP positions go across the bottom on the x-axis. We'll have more to say about that later. And then the height of each point is the negative log of the p-value. So the higher the points, the more significant the association. And conventionally, we've put the different chromosomes here in different colors so that we can easily see where one starts and the other's ends. And each individual point is a SNP, a single nucleotide polymorphism, so a single variant in the genome. Now, there are lots of different pages that tell you about this, but I went to the Wikipedia one straight away because in the first sentence here of the Wikipedia article, it makes exactly the point that I want to start off with, which is that a Manhattan plot is a type of scatter plot. And this is something we encounter quite a lot of in biology and in bioinformatics. So we have these names for specific types of plots, like a Manhattan plot, a volcano plot. Sometimes we talk about a PCA plot. And when we're talking about tools and tooling for doing data visualization, it's worth remembering that these are just special names that we give to different types of scatter plots. So I often get the question of what packages or what tools I would recommend to draw, for example, a Manhattan plot or a volcano plot or something. And my response is always, well, any package that can draw a scatter plot can make you a Manhattan plot or a volcano plot or any of these ones with specific names, as long as we take care to make the X and Y axes right and to have the color scheme that we want. So really, when we're asking about how to draw a Manhattan plot in Python or in any other environment, what we're really asking is how to draw a scatter plot that has this specific pattern of color and these specific X and Y positions. Now, it's always more interesting to work with real data than made-up data, I think, for these kind of little projects. So I've picked an example of some association data that we can look at, and I'm going to bring that up here in the web browser. This is a link to a Nature paper, which is a sort of fairly standard genome-wide association study, often abbreviated as GWAS. And I'll put the link to that in the Jupyter notebook, which, as I say, you'll be able to download from the description. So this study looked at association between SNPs in the genome of humans, and they were looking for association with what they've defined here as risky behaviours. And you can read the description of the paper if you like. There are a few different types of risky behaviours that they investigated. But the data set that we're going to look at is association with speeding. So from the methods, I think the shortest summary is to say that they administered a questionnaire to a bunch of people and asked about whether they ever went faster than the speed limit when they were driving, and then look for association between SNPs and the answer, the way that people answered that question. Now, the data for this particular project is distributed on this link, which I'm going to bring up now. It's distributed via the SSGAC, and on the start of near the top of the data page, this is the link to the various different files that we can download, and you can see there are different types of risky behaviors that they're studying here. But I'm just going to put in directly the link to the data set that we want, which is the speeding data set. Now, we could use pandas to read this directly from the URL, but it's a fairly large data file, so rather than doing that, I'm going to download it, or rather I already have downloaded it here. And if you want to download it and follow along, then again I'll put the URL there somewhere near the top of the Jupyter notebook that I eventually distribute. So to keep things simple, I've just dumped that data file in the same folder as the Jupyter notebook here. And the first thing we need to do is check the format so that we know how to get it into pandas. We'll go through this reasonably quickly because this is not the interesting part of the problem. Because I'm running on a Linux system here, I can be lazy and just use the head tool. This is the name of the file, automobile speeding propensity GWAS. And so this, looking at the first few lines, tells us all we need to know about how to get this into pandas. It looks like it is tab separated, right? I don't see any commas here. We see tabs. The first line is the header line, and this is more or less exactly what pandas expects. So let's immediately go and see if we can get this into pandas. I'll make a new cell down here. We'll import pandas using the usual alias of PD, and then we're going to call the read CSV function. Now, strictly speaking, the only argument we need is the name of the file and then this sep argument to tell pandas that this is tab separated. But when reading in a large file, I always like to just ask pandas to read the first few rows. In this case, let's go for the first 1,000 rows, just to check that we understand the format, check that we don't get any weird errors. If we ask pandas to open the whole file, then it can take a long time to get feedback and see if we've got something wrong. But like I said, this data format is a very simple case, so we have to do minimal wrangling to get it into a pandas data frame. You can see we've got a bunch of different columns. Each row represents one SNP, one variant. And so for each variant we've got a name. Presumably this refers to some known position, known variant in the reference genome. There's a chromosome and a position. There are the base and then the original base. So I think the A1, this column here, is telling us what the effective, what the affecting mutation is. There's a column here which tells us the frequency in the population. So these are all between 0 and 1s. And then the interesting thing here is the p-value. So this tells us how significant the association is between propensity to speed, as measured by this questionnaire, and the particular SNP that we're looking at. So once we're happy with the format here, I'm just going to copy and paste this read CSV. We'll delete the n rows, so we'll tell pandas to read the whole thing. And then I'll remember to store it, and then we'll run that in its own cell. So this is always a good habit to get into when we've got a read CSV or any kind of input that we think is going to take a long time to run. We want to put that in a cell on itself so that we can run it once and not have to re-import the data set every time we want to run some extra little analysis. So that didn't take too long to get in. Let's just check that that still looks okay. So as expected, pandas has given us a default numerical index that just starts at 0 and counts up. And if we look at the size here, it looks like we've got about 10 million SNPs, so about 10 million rows here in this data set, about 10 million different variations. So depending on what you're starting with for your experiment, you'll have obviously different sets of columns and different numbers of SNPs and so on. But you'll probably or pretty much always have a chromosome, a position, and then a p-value. These are the really crucial things that we need in order to make our Manhattan plot. Now, I mentioned earlier that a Manhattan plot is just a special kind of scatter plot. So before we can actually start drawing any plots, we have a little bit of work to do with this. Remember from our description from Wikipedia, or maybe we just already know this about Manhattan plots, what do we actually want on the x and y axis? Well, the x-axis is supposed to be the SNP position, and there are actually two subtly different ways of measuring that. We'll go into that in a second. And then the y-axis should be the p-value, but it's not the raw p-value here. It's the negative log of the p-value, which serves to arrange the points in a way that makes sense, where it sort of compresses the very big scale and also makes the more significant points higher, which is easier to interpret. Now the p-value, we can change this to negative log p-value fairly easily because we're working in pandas. Pandas has tools that make it very easy to apply one expression to a whole column like that. And the easiest way to get the log of something is to use numpy. So we'll import it here. And numpy has a log function that is vectorized. That is to say, it's capable of taking a whole series of numbers and calculating the log of them all in one operation. So all we've got to do is go from our data frame, take that p-value column, so this is our list of 10 million p-values, and pass that to np.log. Okay, so that's going to give us all of our log p-values. We want the negative log p-value, so through the pandas magic we can just put a minus sign at the start and transform those into negative log numbers. And then we want to take this whole expression and store it as a new column, which again is pretty straightforward. We'll just pick a name, let's call it minus log p equals that. And then when we go and look at our data frame, you see we've got this extra new column here at the end, negative log p. Now getting the numbers that we want for the x-axis is a bit tricky because there are two sort of subtly different ways of setting the positions of the SNPs on the x-axis, and I've seen them both used in the real world. One is to simply start counting the SNPs from position 1 and just count 1, 2, 3, 4, 5. So just simply assign a number to each SNP depending on the order that it occurs. That's the simple way of doing it, and the more complicated way of doing it is to use the actual genomic coordinates. So to have the position of the SNP, but rather than counting from the start of each chromosome as we have it right now, starting from the very beginning, the start of the entire genome. So we'll tackle the complicated situation first. We can group these SNPs according to each genome, each chromosome. So if we use group by, pass in this chromosome column, we get a group by object, and then it's fairly straightforward to iterate over that object. Whenever we do group by and then iterate, we get a tuple, we get but the name of the group and then the data itself. So we normally have to write a loop that looks something like this: chromosome, and I often use this word group df for group data frame when doing this, so that we don't get confused between the entire data frame and the data frame just for one group. So if we print the chromosome, you'll see that that should be an in. Let's try again. If we just print the chromosome, these are the labels for each group, and then for the actual data frame we can say take the position and then take the max of that. So this is just going to tell us what's the last position in each chromosome. Right, and so you can see these numbers actually go down fairly monotonically because we order the chromosomes from biggest to smallest. So if we take these numbers, these maxes, then we can add a kind of running total. Right, so let's call this running pos. For running position, this will start off at 0. And then we can put some logic in here to add on this running total each time round the loop. Okay, and now if we print the running position rather than the max here, then you'll see that the first chromosome it's saying starts at position 0 relative to itself, and then the second chromosome starts at this position, this position. So now these numbers go up monotonically because now we're counting the start of the chromosome starting from the start of the entire genome. And there's a nice little consistency check that we can do here. We know that the human genome is around about 3 billion bases. And if we carefully measure the number of zeros down here at the end, we can see that this is about 2.8 billion here. So this feels about right for the position of the last SNP here that is being measured in the human genome. So once we've got this running position here, we can then take this number, or the sort of current running position, and then just add that to the group. So let's take that group df. We'll take that position column and we'll add the running position. Now the output of this is going to be a little bit tricky to see because it's there's a lot of it here, but you can see we're printing chromosome 1, and this is a series now, so these are the positions, or the adjusted positions of all of the SNPs in chromosome 2. So you see the last SNP in chromosome 1 is at this position here, and then the first one in chromosome 2 starts just after it. Right, so we're just adding the running position each time round. So this is where the pandas magic is happening, taking that one integer, this running position, and adding it to the whole position, the whole series of positions for each chromosome. And so finally we need to get all of this into one big series so that we can add it as a new column. There are a bunch of ways to do this, but the easiest way is probably to make a list of these series and then concatenate them later on. So we'll add a list, let's call it cumulative position. Start off as an empty list. We'll, instead of printing this, we're going to append this new series. So at the end, our cumulative position list is going to have, I have to tell Python what I want to actually append it to. Let's try that again. So at the end of running this, our cumulative pos list is going to be a list of all of these series. I know it's tricky to see that, but this is what pandas is telling us here. But the upshot of all of this is that if eventually we use pandas to concatenate this list of all of our series, one per chromosome, we should hopefully end up with one big series. So here it is, one series that has all of these adjusted or cumulative positions. And now we can go ahead and add that as a new column. So we'll just make a new column called cumulative positions. And as usual, once we've done some calculation, we've added a new column, we'll look at the data frame so we can see that now we've got this cumulative position at the end, which is kind of the the genomic coordinates not relative to the start of the chromosome, which is what pos is, but relative to the start of the whole genome. Hopefully that made sense. It's a fairly complicated calculation that I had to go through quite quickly. Now, I mentioned there were two different ways of, or two different things that we could put on the x-axis. One is this cumulative position. The other, much simpler way, is just to number the SNPs 1, 2, 3, 4, and have that sequence going along the x-axis. And that is actually very simple to do. We already have numbers that go 0, 1, 2, 3, right? They're the index, they're the default index that we get. So we can get that with the index attribute. Remember, this isn't a method, so we don't have to have parentheses, but we can just make a copy of that and put it in a column. So let's just call it SNP number for now. Store a new column. And so now in our data frame, these last three columns are the interesting ones. These are the ones that we're going to use to build our Manhattan plot. So we've got negative log p, that's going to be on the y-axis, and then we can try putting each of these different columns on the x-axis, and we'll see, hopefully it'll be easier to see visually once we have a visualization, what the actual difference is. So now we've done this preparatory work, we've done the calculations and taken advantage of pandas, vectorization and so on to do these. Notice that we haven't had to write any loops really, apart from this loop that goes through the chromosomes, but we have avoided writing any loops that iterate over each line, which is a good thing because there are 10 million of them. So now we can get set up to do some visualization. So we'll import seaborn, which is my favorite charting package. Seaborn is built on top of matplotlib, so we're actually going to be using both of them as we go through this. And because I'm working in Jupyter, I need to remember to put in the inline command here so that I can actually see the charts. So in seaborn, which we've given the alias here SNS, the tool for building a scatter plot is relplot, short for relationship plot. So we'll do relplot. And let's start off, we're just going to call it with our data frame and say that on the x-axis we're going to have this cumulative position that we created earlier. And on the y-axis we're going to want the negative log p column. So I'm going to hit go and we'll let Seaborn think about that. Now you'll notice that this chart is taking quite a long time to draw, right? And that's because it's trying to plot 10 million rows. Remember, the nature of Manhattan plots is that they're scatter plots and each point is one SNP. We've got 10 million SNPs, so we're asking Seaborn here, or matplotlib, to draw a scatter plot with 10 million individual points. So it's not surprising that it's going to take rather a long time to draw. I'm actually probably going to pause the video here for a few minutes. Oh no, I don't have to. Okay, so it didn't take a super long time, but still longer than we would like, especially when it comes to starting to iterate on the chart. So before we do anything else, I want to make it so that we can draw a version of this chart very quickly, which means that it will make it much easier to then do all of the fixes and add all of the modifications that we want. Now, the easiest way to do this is to just plot a subset of the data, and we've got to be a bit careful here. We don't just want to do something like this and say plot the first 1,000 points, because obviously the first 1,000 points are all in chromosome 1. So if we do this, we're not going to really get a representative chart of how our whole data set looks. A much better idea when we want to speed up a plot by plotting a subset of the data is to use sample to randomly sample some points. So let's just ask Seaborn to plot 1,000 random points here, and the chart still doesn't look good, right? It's still not a Manhattan plot, but it does at least plot very quickly, so this will make it much faster to improve on this plot and iterate it. Now the first thing we need to do, I think, is probably fix the aspect, right? We want this to be a wide plot that takes up more of the space that we have available. So let's set aspect equals 4, so we'll say make it 4 times wider than it is high. Right? That looks like a good kind of shape. And the size here, I don't think we need to adjust. This text is reasonably easy to read on this the monitor that I've got here. Hopefully it's reasonably easy to read on the video as well. So here's a scatter plot. Now it looks like this is not really going to give us enough information, enough points. So let's plot 100,000 SNPs. That's going to be more dense and start to look a little bit more like a Manhattan plot. Now, what do we need to do the next set of jobs? Probably the next job is to make the thing coloured by chromosome. Right, so the whole nature of what makes a Manhattan plot is that the chromosomes are different colours so that we can easily see the distribution of these points. And because we're working in seaborn, this is very easy. All we've got to do is go up here to the code here where we're calling relplot and set hue, which is what seaborn calls colour, and we'll say make the colour of each point determined by this CHR column, this chromosome column. So our plot will take a little bit longer to run. And now we run into something, and this is one of these interesting points that I mentioned at the start of the video, where an interesting fact about seaborn, a slightly surprising behaviour, is that if we ask it to colour something that is numerical, right, so our chromosomes here are numerical, then by default Seaborn is going to pick a numerical palette, what it calls a quantitative palette, in other words, one that goes from a pale colour to a dark colour. And that is not going to make it easy to see the divisions between chromosomes. Now, in a way, this is a bit of an accident, right? It's kind of a coincidence that this happened because we happen to have, in this data set, only the autosomes, so only the chromosomes from 1 up to 22. We don't have X and Y. If we had X and Y in here as well, then when pandas read the data file, it wouldn't have assigned this as an integer type, it would have assigned it as a string type, or rather an object type, and we might not have run into this phenomenon. But in any case, if we know how to use Seaborn, it's pretty easy to fix. We can just explicitly set a qualitative palette, a categorical palette. So let's try that. Let's just pick set1, which is one of the built-in Seaborn palettes that works perfectly well for medium numbers of categories. And so you can see we're gradually starting to approach something that looks more like a Manhattan plot, right? We've got our data divided up into individual chromosomes. We've got this kind of ugly legend here that we're going to get rid of soon. And then we've got, this is our cumulative position. So we're going to fix this in a minute. We're going to change these X labels and X ticks to reflect the chromosomes. We'll get rid of this legend and we'll play about with some different colours. Before we do all of that, though, I just want to take a minute to copy this and run the same code with the alternative x-axis. So this is the version of a Manhattan plot where on the x-axis we've got the cumulative position, right? So this is actually the genomic coordinates, and we can see things like gaps, right? So there's a region here in chromosome 1 where there aren't any SNPs. I'm pretty sure this is the centromere, but if anyone knows any more, if I'm wrong, then please mention it in the comments down below. Let's see what happens if instead of using this cumulative position we do the simpler thing, which is just to use the SNP number on the x-axis. So all I'm going to do is copy and paste this relplot call, and instead of saying x is the cumulative position, we'll ask for the SNP number. Now there are going to be some random differences because we're still randomly sampling here numbers, but you can kind of see the pattern going on here, right? In this version that just numbers the SNP 0, 1, 2 according to their order, we don't see any gaps, because if there aren't gaps there won't actually be jumps in the sequential numbering. And like I say, I've seen it done both of these ways in real life. So if you're interpreting a Manhattan plot, or if you're choosing to make one yourself, you have to either know which version you're looking at or choose which version you're looking at. So either way, I think we're going to pull out the same actual information, right? So you can see over here in this pink chromosome, I think it corresponds to chromosome 17. There's an area here of SNPs with very high association scores, and then the same thing here. It just looks slightly different. Okay, now let's start tweaking this chart a bit. I think we're going to work with this one. So I'm going to leave both of these charts here in the notebook, but I'm going to copy this one. I think this is probably the more common way to do it. So I'm going to copy this version that uses the genomic coordinates, and we'll see what modifications we need to make to get this looking a little bit better. One thing that we nearly always need to do in seaborn is when we've got very large numbers of points, we normally want to turn off the outlines, right? You can see that each point has this little outline, so we can easily turn that off with line width equals 0. We'll tell seaborn not to draw the outline. And the other thing that usually works very well in conjunction with that is to turn down the size. So we can do that with the S argument, which will tell seaborn how big we want to make the points. Now we can, there's nothing particular about a size of 4. I'm not even sure what units these are in. So we can just adjust this until it looks right for our use case. And this legend here isn't really telling us very much. In fact, it's quickly going to become irrelevant because we're about to label these explicitly on the x-axis. So let's turn it off because that's an easy fix. We can just do legend equals none. So you can see the pattern. This is a very typical way of using seaborn, right? We start off with just the most important bits, right? That bit that I've highlighted there is the sort of minimal amount of information to do the scatter plot, and then we can tweak it in these various ways by setting the colors and the palettes and adjusting the way that various different things look. So the next big job then is to fix the x-axis labels. We need to put the actual names of the chromosomes here, and we need to make sure that they appear in the right place. Now in order to do this, we need to call some methods on the chart object. So we have to adjust this so that we actually store the result. So this G I've used as the variable name because G is short for grid, because what we always get out of relplot is a facet grid. Again, you can read about all the details of this in the book if you're interested. But the upshot of it is that relplot always produces a grid because often it's used to make small multiples, and there's a special case which is if we have a grid that has just one chart, which we do here, we can use this special attribute AX to just get the axes. So now we've gone inside the grid, we've got hold of a reference to this axis object, and this is the object on which we can call the various other different methods. So let's start getting this x-axis looking the way that we want. We need to set the label, and it's going to be something like chromosome. That's job 1. So you'll see the previous it used to say cumulative pos because that was the column we were using, but we actually want it to say chromosome. And then we want to set the ticks, and this is something that is very commonly confusing to everyone when they start using matplotlib, which is that there's separate methods for setting the tick positions and the tick labels, in other words the text. So we need to remember that in the matplotlib terminology, the x ticks are the little vertical lines that come down from the axis line, and the x-tick labels are the text that gets displayed. So we need to come up with a list of positions where we want the ticks, and then a list of labels that we want to be applied to them. And this is another case in which pandas is going to come to our help and make this not too painful to do. So to find the positions, we want the midpoint, right? We want each chromosome to be labelled more or less halfway along its length, right? So for this purple chromosome here, right, which is 1, 2, 3, 4, we want the chromosome 4 to be about where my cursor is there. And we can easily get these positions by taking our original data frame. We can group it by chromosome. So we'll treat each chromosome separately. Within each group, we can get this cumulative position. And then we can take the median. The mean would also probably work fine because these are densely distributed. But this gives us a series, which is the median position for each chromosome, which happens to be exactly where we want to put our labels. And this isn't actually a list, this is a pandas series, but a nice thing about pandas series is that they will usually behave like a list in most contexts. So I can just copy this expression, we'll pretend it's a list of the positions, and we can plug it into the method that sets the tick labels. So let's try that, set ticks, and remember we're just telling seaborn here, or rather we're just telling matplotlib where we want the labels to hit. Okay, so if we ignore this big chunk of output here, which is telling us that this got this list of tick objects, you can see that our ticks now appear just where we want them, right, in the midpoint, more or less, of each of these coloured blocks. Now, although we've told matplotlib where we want the ticks. We haven't told it what text we want, so that's the next job. And that is, in fact, even easier to get. So if we just get the list of chromosomes, and then we'll get the unique ones. Right, that's going to give us a nice list that just goes from chromosome 1 to 22. And we can call another method to set the labels. So set x tick labels. Let's see what this gives us. Now I have made some typo here, so there should be g.ax. That gets us the axis, and then that calls the method. Let's try again. Again, ignore all of this output here. Okay, and now we're looking at something that looks a lot more like a Manhattan plot, because we've got the ticks in the right positions and they're labelled according to the chromosome. So this is just what we wanted. And we already turned the legend off. So we're gradually building this up. Again, this is a very typical pattern that we see in when using Seaborn to do fairly specialized charts like this. We've got one big function call to relplot to set all the things that we can set in Seaborn, and then we've got a bunch of extra little method calls here to adjust the properties of the chart that we can only adjust by going through the matplotlib interface. So we can see nicely the distribution of chromosome sizes and SNPs and so on. I'm going to leave this code then, I think, in the notebook. Actually, let's do one more thing before we leave that and declare it done. Let's just put a title on here. It's usually a good habit to get into. So in this case, we need to go from the grid to the figure, and then we need to set a super title. So let's just call it something to do with GWAS. Think of a title like, let's say this, GWAS plot. Let's see how that looks. Okay, yeah, we can leave it like that for now. So that's going to be our sort of version 1 or variation 1 of our Manhattan plot. And if that's all that you wanted to see, you can quite happily leave the video here and go and grab this code from the notebook, which is linked in the description, and go and use it. But if you stick around, then we can try a few different variations of on this theme. Why are we actually bothering to do this in Python in the first place? There are actually lots of tools that will build Manhattan plots with probably a lot less effort than this. Well, the reason that we want to do this in pandas and Python is that it gives us a much more control, right? We can control exactly how the various aspects of the chart look and work in a way that we might not be able to with a standalone tool. So I'm going to copy and paste this whole chunk of the code, and we can look at a few different variations. One obvious thing is to change the color scheme. So sometimes we want to have the chromosomes all in different colors like this. Sometimes we want them to be in the classic kind of alternating black and gray. And so that's not too tricky to do. We can just set this palette here. We're allowed to do this in Seaborn. Rather than just giving the name of a color scheme, in this case set 1, we can give a list of colors that we want to use. Now if we want it to go grey, black, we have 22 chromosomes, so we need it to go grey, black 11 times. And one useful lazy trick that we can do here is just to make a little list of grey and black, and then multiply it by 11, and this will give us a list of 11 times the strings grey and black. So see what happens if I copy that expression and put that here into my palette. Then we're going to get a version of the plot that has grey and black rather than the individual colours. And of course we can pick whatever colours we want for these types of things. Once we start playing about with colour though, we might realise that we have a lot of flexibility if we're allowed to say exactly how we want to divide up the colour, and then specifically what palettes we want to use. So if we adjust, let me show you another way of dealing with the colour. So one is to explicitly make a new sort of colour group column, and we can do that by taking the chromosome and then applying something to it. So let's start with this chromosome column, and let's say we're going to make a new column which is going to have one value for odd chromosomes and one value for even chromosomes. It doesn't actually matter what the values are, so I'll just call them A, B, C, etc. for now. Probably the easiest way to do this is with apply, so we can write a little lambda expression here that says, well, for each of these values we want the letter A if the value divided by 2 or modulo 2 gives us a remainder. So this is the way of checking to see if a number is even. And if it's not, then we want the value B. So this is just going to give us a new series that is B for odd numbers like 1, 3, 5 and so on, and A for even numbers like 0, 2, 4. We don't actually have a chromosome 0, but you get the idea. And I can call this, let's give this a new column, color group. So this column doesn't actually contain colors, right? It's just got these groups A and B. So what does this allow us to do? Well, let me copy the code down here. For one thing, it means that if we say that we want the hue, the shade of each point to be determined by this color group column, then we don't have to bother repeating things. We can just say, well, there are two color groups here, A and B, and so we only need to give two colors here. Just for a variation, let's do red and pink, just to prove to ourselves that we can pick any colors. So that's how we get that kind of striped effect. It means the palette can be a lot simpler. It also means that by changing this color group, we can very easily highlight particular points in a very flexible way. So let me copy this little bit of code here. I'll make a new cell down here. And let's try this. We'll make this color group column just as we did before, but then we'll grab just the rows belonging to chromosome 3. The easiest way to do this is lock. So this expression here will find all of the rows for chromosome 3. We can then grab the color group column for all of those rows. So that's the series of all these B's. And then because I've used lock here, we're allowed to assign it. So we can say that, give that C. We'll look at our data frame here. So now this color group goes B, A, B, A, B, A, apart from in chromosome 3 where it goes C. Now, the whole reason that I'm going through all of that is so that we can then copy this code. Right. So if we have 3 different values now in color group A, B, and C, we're allowed to give 3 different, or rather we have to give 3 different colors here. So let's do red, pink, and blue. And so you see it makes it very easy to highlight one particular set of points in a different way. Here's another variation on the same theme. Let's say we want to highlight just a particular region of a chromosome. We can be as specific as we like, as long as we can come up with some expression here that defines which points we want to color differently. We can write it. So let's try and do a slightly more complicated filter. We'll do chromosome, I don't know, 6. Doesn't really matter what we pick. So let's find chromosome equals 6. We want the rows where the chromosome is 6 and the position, let's say less than 5. Let's make it 50 million. All right. So hopefully this is going to highlight. So it's going to change just the color group of just chromosome 6, just this region here between 0 and 5 million. And again, we can plug in any arbitrary sort of filter here to draw attention to particular points or particular snips in here. The other thing, to take this to the logical conclusion, is that we could do this not with a filter but with a list of snips, a list of positions that we are interested in for some other reason, right? Maybe they've been previously implicated, or they're all part of some pathway or something. Let's try it. If we let's get some marker names. and we'll randomly sample 100 of them, for instance. So this is a random sample of 100 marker names. We'll call this my markers. Let's imagine the real world, rather than randomly sampling them, of course, we've got a list of marker names in some other way. Well, if we copy all of this code like we did before, and then the filter here to set this color group C, rather than doing anything to do with the position, we'll just set this on rows where the marker name is in this list of my markers. I have to be careful to spell that right. Okay, and so now you can see we've got the chromosomes are all going pink, red, and then sprinkled in here we've got these blue points, which are the ones that we pulled out randomly here from this name. Again, this would be a more convincing example, I think, if we did it using points, or rather marker names that we'd got from some other source. One other thing that we very commonly see in these kind of plots, in Manhattan plots, is it's often traditional to draw a kind of horizontal line to represent some aspect of, or some cutoff rather, of significance. And we can do this easily again with the axis. So there's a H line, horizontal line. And all we've got to do really here is put in where we want the horizontal line. So let's say we want negative log p-value of 12 as our cutoff. We can throw in AXH line there and it'll draw a line. And whenever we're putting lines on charts, it always feels a little bit stark to have it as an unbroken line. So I normally throw in line style and pick something that involves dots. And also this is a bit big because the points are so small. Having such a thick line here doesn't look quite right. So let's turn the width all the way down and we'll just make a very light little line going across. I think that looks a little bit more elegant. Once we've got this line in place, another obvious thing to do is to change the colours of just the points that lie above the line. And we could do that fairly easily, again, just by changing our filter up here. Now I can't resist showing you that because we're working here within seaborn, all the work, the initial plot is being generated here with relplot. So it's actually very easy to do quite a lot of different variations of this. Let's copy everything here until the plot itself. I'm going to put this in a new cell down here. And let's say we want to change this into a small multiple plot. Let's say we want to do a separate Manhattan plot for each of several different categories. Well, we can easily add it down here. So if we just say, for example, row equals, let's have a different plot for each of the different bases. We can say do a separate row for each of these, the values in this A1 column. And just with that one line, we get the whole plot here repeated for A, T, and C. These are the various different bases that these SNPs are found in. I'm not going to try and draw any biological inference here, but I think it's worth just showing you that because we've got this flexibility, we can do that very quickly. We do have to make some adjustments to the chart if we're going to do this small multiples trick, because to set the X ticks and the labels and the horizontal lines and so on, previously we could do that just on this special g.ax attribute. This won't work down here because now we have not just one axis, we've actually got three different of the axis objects. So we have to explicitly iterate over them, but that's not too terrible. We can take this g.axes.flat. This will be a flattened list of all of the axes, and then I can copy all of this code, make sure it's properly indented, and if we just run this on each axis individually, then that should be enough to apply this same styling with the setting the chromosome names and the X-tick positions and all the stuff that we did before, and doing all of that on these separate axes as well. Now, one last variation that I want to show you is something that we see, again, fairly frequently in the real world, which is taking a particular set of markers, a particular set of positions, and annotating them with their name or some other extra bit of information. So I think this is going to be easiest to see if we go back to a fairly simple plot. So let's take this version of the plot, which is just grey and doesn't have any lines or anything clever going on. Excuse my wild scrolling. I'm going to paste this in here, and we'll just look at a couple of options for putting labels on here for annotating these with individual names. Now, this is going to be easier to do if we take this sample and we'll give it a specific name. So I'm just going to make, for convenience, a data frame called my data, which is going to be a random sample of 100,000 points. If we do this, and then everything else works on my data, I think that should still look okay. All right, and now what we can do, and I'll work in a little cell just above here, is like we did before. We have to choose which points we're going to annotate, and the simplest thing to do is probably just to pick ones that are above some threshold. So let's get our negative log p column. We'll find the values that are bigger than, let's say, 20. We can adjust this cutoff, of course, later on. And if we plug that back into the data frame as a whole, these are all of the markers, all of the positions in this random subset of the data, where you see this negative log p column. These are all the ones that are greater than 20, so the strongest association, the most significant. And what we can do is now iterate over all of these, and for each of them call an annotate function. So once we have our matplotlib axis object, we can call annotate and tell it to put a particular bit of text at a particular position. So let's put all of these together. Here's the selected markers. I'm going to come down here and do this is the very last thing. Okay, so this draws the plot and then produces, it does this filter, gives us our data frame here of all the points that are greater than 20. And then to actually run some code on that, all we've got to do is call the apply method, write another lambda expression. And so if we just do marker name, for example, we have to remember that we're doing this over rows, not columns. Then this will just give us a list of all of the marker names. But if we actually change it to annotate, then we can say draw the marker names on the chart. So let's see how this might work. We have to do g.ax to get hold of the axis object. Then we call the annotate method, and it has first argument is the text we want, which in this case is the marker name. And then the second argument is this tuple, so we need to give a tuple of x and y positions. I'm just going to close that method here before I lose track of my parentheses. So marker name is the text, and then the x position is just going to be the cumulative position, and the y position is going to be the minus log p. So just exactly what we had for the x and y up here. I'm actually going to change the argument here in this lambda. Let's call it p for point, just so that we don't get confused between x as a variable name and x as the x position. Okay, so find the points that we want to annotate. For each point, run this annotate method and give it the name and the x and y positions. Let's see what happens when we do that. So the output, we get this big long list of annotations. But if we go down here to the chart, you'll see that it's now drawn on the chart for all of the points that are above 20, their actual names. And here, of course, we got this marker name. This could be some other bit of metadata. As long as it's in the file, we can get hold of it with a filter. Now, we've got a very common problem here, which is because the whole point of doing Manhattan plots often is to show regions where we have dense clusters or dense areas, regions of very strongly associated SNPs. We've got this problem where inevitably we end up with a whole bunch of labels that lie on top of each other. And I wanted to just finish off by showing you quite a cool package. I have nothing to do with this package. I just sort of stumbled across it when I was trying to solve this problem, called adjustText. Let me bring up the GitHub page here, and again I'll make sure this is linked from the Jupyter notebook. Here's adjustText, and this is a library, a package that will solve exactly this problem we've got here. It'll take a set of annotations on a matplotlib chart like this that all overlap, and will move them around so that they don't overlap. So the way to do this is pretty straightforward. I'm just going to copy and paste some code that I have off screen. So we're going to import this library. I've already installed it, but that's very easily done. And we have this method called adjustText. And then in order to use it, what we have to do is take the result of this, which is going to be our list of annotations, ask pandas to give it us as a list. If I just run this code, I always forget it's actually called toList. So this gives us a list of text objects. We can tell this is a list because it starts and ends with square brackets. We'll call this annotations. The last thing we have to do then is pass this list of annotations that we've made to the adjustText function that we just imported. Now you might notice when I run this it takes a little bit longer to draw than the previous charts, because I'm guessing this library is using some sort of genetic algorithm or something in order to figure out the best way to lay this out. But you'll see it does a good job of moving these annotations, these bits of text away so that they don't overlap. Now it does introduce another problem, which is that where we've got very densely clustered points like over here, this is all now surrounded by a sort of big blob of text annotations. By moving them away, there's no way to tell which points they belong to. But it does also have this rather neat ability to draw arrows. So let me show you what happens if we add an arrow props dictionary. We can say that we want to draw arrows going from the annotations to the points themselves. We can draw a little ASCII text arrow here, and this adjustText library will take care of drawing the arrows. So you'll see it draws arrows pointing towards the various different points, and we can even highlight them a bit. Let's say that we want these to be in blue, for instance, just to make them stand out a bit against the background. So moving the text around introduces one problem, but happily the library kind of has a solution. And again, this would be a more convincing example if, rather than just randomly picking a set of markers to highlight, we got them in a more structured way. So that, I think, is where we're going to leave it for now. And thanks for sticking with the video for this long if you stuck all the way along to the end. Don't forget that if you have any questions, if there's anything that wasn't super clear, feel free to just leave a comment down below and I'll try to get back to you. And if this stuff seems interesting or useful, then do go and check out the Biological Data Exploration book, which will be linked in the description as well. That has loads more detail on all the stuff that we've been working on here, and also you can get it with a bunch of other videos if you like the style thing. If you want to use any of this code for your own work, then make sure you click the link to get the Jupyter notebook. I'll just stick that up on GitHub so you can run it and tweak it and just reuse the same code for your own work if you like. And if you have an interesting biological data visualization problem or question, then feel free to drop me an email. I'll put that in the description as well, and maybe it'll find its way into some future video. Lastly, if you're interested in stuff like this, then you can sign up for the Python for Biologists newsletter and or follow the account on Twitter, both of which will get updated when I make more videos like these, which hopefully I'll do in the near future. So thanks for watching, and I'll see you soon.